Microbiology Assignment Help. For any assignment-related queries, call us at +1 678 648 4277. You can mail us at info@nursingassignmenthelp.com or reach us at https://www.nursingassignmenthelp.com. What does proton motive force mean, and why is it important in biology? Answer. The proton motive force is an energized state of a membrane created by the expulsion of protons to the exterior face of the membrane, creating a charge separation similar to a battery. The dissipation of this charge gradient through a membrane-bound ATPase complex provides energy for ATP synthesis. B. How is rotational energy in the ATPase used to produce ATP? Answer. Rotational energy in ATPase is used to produce ATP via proton motive force. Problem 1. Answer. Rotational energy in ATPase is used to produce ATP via proton motive force coupled to conformational changes in subunits of ATPase holoenzyme. Movement of protons through the FO subunit drives rotation of the C proteins, generating torque. Conformational changes in the beta subunit transfers this potential energy to the F1 subunit. A. What are the differences in electron donor and carbon source used by Escherichia coli and Acidithiobacillus thioporus, a sulfur chemolithotroph? Problem 2. nursingassignmenthelp.com. Answer. Electron transport chains with oxygen as the final electron acceptor are utilized by both organisms. E. coli oxidizes organic compounds to produce energy, whereas A. Thioporus oxidizes inorganic sulfur to use it as an electron donor. B. The following is a series of coupled electron donors and electron acceptors. Using Figure 5.9, order this series from most energy yielding to least energy yielding. C. Explain the circumstances under which the same substance can be either an electron donor or an electron acceptor. Answer. The coupler pairing determines acceptor versus donor. The substance with a more positive EO is the E donor and the more negative EO is the E acceptor. Thus, depending on the available copule for reduction or oxidation, and the relative concentrations of each species, both E donor and acceptor, a substance will act as an E acceptor or donor. D. Consider the following reaction. NADH plus fumarate succinate plus NAD plus H. nursingassignmenthelp.com. A. NADH plus fumarate succinate plus NAD plus H plus using Table 1 calculate the delta E0 of the reaction. What is the delta G0? Does this reaction produce or consume energy? B. Does this look to you like a potential reaction in a respiratory pathway? Why or why not? Answers will vary. Problem point 3. A. Explain the following observation. Cells of E. coli fermenting glucose grow faster when NO3 is supplied to the culture, and then grow even faster when the culture is highly aerated. CH5 application question 4. Answer. When E. coli is growing in a fermentative mode, there is no external electron receptor and the energy yield is low. When NO3 is added to the culture, nitrate acts as an electron receptor. Under these respiratory conditions, the energy yield is greater than fermentative conditions, but still less than when O2 is the final electron receptor, aerobic. B. Discuss why energy yield in an organism undergoing anaerobic respiration is less than that of an organism undergoing aerobic respiration. nursingacgnmnethelp.com. Answers will vary. Basically, O2 has a more positive EO. Therefore, in coupled redox reactions, more free energy is available using O2 as the terminal electron acceptor. Problem point 4. Explain our current understanding of molecular adaptations to the cytoplasmic membrane that are present in psychrophiles and describe their habitat. How about thermophiles? Answers. Typically, psychrophiles are found where the temperature is constantly cold opposed to seasonal winters. Their cytoplasmic membranes contain higher concentrations of unsaturated fatty acids and sometimes polyunsaturated fatty acids that maintain a semifluid state at low temperatures. Enzymes of psychrophiles are cold-active with greater amounts of alpha helices and fewer beta sheets, as well as greater numbers of polar amino acids. Thermophiles would be found in environments that are consistently hot. Therefore, thermophiles have enzymes and other proteins that are more stable and function optimally at high temperatures. Cytoplasmic membranes of thermophiles also contain lipids rich in saturated fatty acids, which allow form stronger hydrophobic environments, more stable membrane in heat. nursingacgnmnethelp.com. Problem point 5. A. Describe the growth cycle of a population of bacterial cells from the time this population is first inoculated into fresh medium. Answers. The 4 stages are lag, log, exponential, stationary, and death phases. Measurements of total versus viable cell counts will be identical during log phase growth. However, during stationary phase, the viable count may differ from total cell count, since intact cells that are no longer viable will be included in the total cell count. B. Describe a direct and an indirect method to measure microbial growth. Answers. Direct counts can be accomplished using a light microscope. Medium containing the cells is placed in a counting chamber, which is a microscope slide containing etched lines forming a grid of known size. Because the cover slip is a known distance above the slide, each grid contains a known volume of medium. By counting the cells in several grids, the number of cells in the culture can be calculated. Indirect counts are based on turbidity of the medium, culture. The culture is placed in a spectrophotometer cell and the absorbance is measured. Under limited conditions, log growth, the number of cells present can be estimated fairly accurately using a standard curve generated by plate counts at the time of absorbance readings. nursingacgnmnethelp.com. C. If a 1-liter culture of rich medium is inoculated with 3 bacterial cells per milliliter, how many cells will be in the culture after 2 hours, given that there is an hour lag phase and the generation time is 20 minutes? After 3 hours? After 4 hours if one of the initial 3 cells was dead? Answers. Problem point 6. As part of a UROP project, you get a chance to quantify bacterial numbers in environmental samples. The graduate student you are working with has used centrifugation to concentrate 10 1-liter samples of oligotrophic lake water 1,000-fold, i.e., in a final volume of 1 milliliter. She asks you to determine the number of viable bacteria in the samples. By plating 10-fold serial dilutions on minimal media and incubating in ambient O2 at 37 degrees Celsius, you get small, slow-growing colonies. nursingacgnmnethelp.com. Starting dilution, 1 milliliter, amount spread on plate number of colonies concentrated sample 100L2 numerous to count 10 to 1 100L 50 10 to 2 100L 5 10 to 3 100L none 10 to 4 100L none 10 to 5 100L none. When you plate and incubate on rich media using the same conditions, you get no growth at all.